Hello, my name is Joe, and today in this short video I'll be discussing the role the adenosine deaminase protein, or the ADA protein, has in the human body, and in particular I'll be discussing two chemical reactions that it catalyzes. First of all, we'll be looking at the chemical reaction of adenosine into inosine, and as I stated, this is catalyzed by the ADA gene and is associated with the removal of the NH2 amino group and the addition of the oxygen group in place of that. In normal functioning cells with active ADA protein, we have very low levels of adenosine, as adenosine will be readily catalyzed into inosine by the ADA enzyme. However, in cells where there is a loss of function mutation in the ADA gene, we will see the inability to catalyze adenosine into inosine, and as such we'll have significant build-up of adenosine in these cells. The build-up of adenosine is then associated with the dysregulation of the immune system. It is associated with lymphocyte apoptosis, which is essentially death of highly functioning white blood immune cells. And we also see inhibition of those lymphocyte activation as well. Okay, and so now we will be looking at the second chemical pathway that is catalyzed by the adenosine deaminase enzyme. So in healthy cells, we have deoxyadenosine that is catalyzed into deoxyinosine by the ADA enzyme, which is also associated with the removal of the amino group and replacement with the oxygen, and it differs from adenosine as it has lost an oxygen here, and likewise deoxyinosine differs from inosine as it's lost an oxygen on the carbon ring there as well. In cells where the adenosine deaminase enzyme is defective, where we've got a loss of function mutation in the ADA gene, we are unable to catalyze deoxyadenosine into deoxyinosine, and we have the generation of this new molecule down the bottom here known as deoxyadenosine triphosphate. As a result, we have significant increase in the levels of this deoxyadenosine triphosphate, and that is associated with defective lymphocyte activation and lymphocyte toxicity, which essentially will decrease the overall effectiveness and functioning of the immune system, particularly the adaptive immune system.